Hi, everybody's on mute, just so you know. My name is Agnieszka Carpenter, and I'm the Executive Director of BioME. Welcome to BioME Student Showcase. This is our very second student showcase, and we spend the first part of the day talking to high school students. So now we're excited to see you all on our Zoom meeting, our virtual event. And I will start with just a couple slides of introduction, and then we'll proceed with our first contestant. I'll go over some rules just to make sure everybody is aware of what we're doing here. So we're going to go between 4 p.m. and 5:15. I should also say hello to all of the contestants, all the mentors who are joining us today, all the 6 judges we have on this call, and some of our BioME board members. And I hope everybody can see my screen. These are all of our contestants today. You may know each other or maybe not. The schools represented today are University of Southern Maine, UMaine, Colby, and UNE. And we'll be going in this alphabetical order by last name. Just to remind you what you're competing for, these are the prices for first, second, and third place. So the stakes are pretty high. The highest prize today is $2,000. Our judges today, we have 6 judges. We have Dr. Ann Brescia from MMCRI. We have Dr. Christy Townsend from University of Maine. We have Gary Goodrich, owner of Replinova, who used to be the president of our board. We have Patrick Breeding, project manager and co-founder of Dermaris. Dr. Stephen Polsu from BBI Solutions, and Dr. William Palin, who's the chairperson for the biotechnology sector of Maine Technology Institute. And thank you to all the judges for joining us today. They'll be asking you some questions after your presentations. Just to go over the rules before we start. So you have 3 minutes and 5 slides to give your pitch. We will be timing you. So basically when I say, you can now start your presentation, you just go, and Bree will be timing you. If you go over, we might say your time is up. You will also be sharing your screen, and we will not start timing you until you start sharing your screen and talking. So don't be stressed if something's not going well. We ask that your camera is on. We ask that you unmute yourself when you're speaking. I think everybody knows how to share their screen. And after the 3-minute pitch, there will be 2 to 3 questions from the judges. Also, when you are not speaking, please make sure to keep your microphone on mute, just so we don't have any background noise. And the winners will be announced tomorrow at noon. I wanted to wish everybody good luck, and thank you for joining us today. And I will stop my share right now. Okay, does anybody have any questions before we start? No. Great. Okay, so our first contestant today is Rachel. Yes, Rachel, do you want to unmute yourself? Hi, Rachel.
Hi, can you hear me okay?
We can.
Fantastic.
If you want to share your screen, we'll be able to see.
Sure, just give me one moment. And get everything set here. Sorry about that. I have everything set up from when I was teaching earlier today, so my screen is a little bit. Can you guys see the slides okay?
Yeah.
Fantastic. All right, just give me 1 second.
Sure.
All right, ready whenever you are.
Okay, why don't you start now?
All right. So astaxanthin, a red carotenoid, is a powerful antioxidant used extensively in aquaculture, nutraceutical, and medical industries. And so one of the most potent antioxidants in nature, astaxanthin has shown significant in vivo and in vitro protection from photooxidation, anti-inflammatory abilities, and enhancement of cardiac and immune response. Astaxanthin is also essential to crustacean and fish growth, and yet with all this clinical and commercial significance, there are still major issues with production efficiency in this billion-dollar market. Hematococcus pluvialis is a freshwater green microalga with the ability to produce the highest concentrations of astaxanthin. This pigment is accumulated in lipid droplets in the cytoplasm during a planospore formation and after a stress response, such as excess light. Astaxanthin is believed to then play a key role in preventing further oxidative damage and loss of structural integrity to the photosystems of the cell because these same stressors cause an increase of reactive oxygen species. Several cellular components act as ROS and redox sensors within the cell, such as elevated reduction of plastiquinone to plastiquinol within photosystem 2 leads to an increased transcription of 4 out of the 5 main genes in this pathway. Current research also suggests both post-transcriptional and post-translational control of these carotinogenic genes by increased ROS concentrations. And even with this vast wealth of knowledge pertaining to astaxanthin biosynthesis, there are still massive gaps of knowledge within this field. And while ROS has been clearly established as having a role in this cascade, the magnitude and importance of that role has not been clearly defined. And so the overall goal of this study that I'm undertaking is to analyze the magnitude of importance of ROS on the astaxanthin biosynthesis pathway within the microalga H. pluvialis. And to quantify this relationship, this study will be analyzing the following: ROS cellular production to astaxanthin synthesis over a series of environmental stressors, as well as production of astaxanthin after the addition of an ROS-producing enzyme to isolate this ROS impact, as well as addition of an ROS scavenger to an encysted culture. Data analysis will be completed via dry weight and HPLC analysis, fluorometric assay of an ROS-sensitive dye, and a linear regression analysis. And so this model, with emphasis on the addition of an ROS-producing enzyme and ROS scavenger, is novel in its approach. And so from this study, several short-term and long-term outcomes are expected. These include increased knowledge of the magnitude of importance of ROS in this pathway, improved understanding of the purpose of astaxanthin, and increased efficiency of astaxanthin production on both a laboratory and commercial scale. And so though this project may seem small in scale, the study has the possibility to affect large-scale changes through collaboration with industry members and also by adding novel data to the field. Thank you very much for your time.
Excellent. Thank you so much. And we'll take the first question.
Thank you, Rachel. That was great. So I think you touched on it on the first slide, but now that I've seen everything, if you could just talk again about what the commercial applications would be if you increased the biosynthesis of this compound.
Absolutely. So this isn't just a medical industry. There is a ton of interest in cardiac as well as active use in medical clinical trials. But there's a massive amount of industry for the money aspect is in the nutraceutical and the aquatic fields. So nutraceutical industries, there's 5 major ones in the world that produce most of this. I actually am able to work with one of them in Iceland, and they are producing the majority of the money that's coming in for this. You can actually buy astaxanthin in the store. Just you and I could walk into a Walmart or a CVS. There's a lot of interest in kind of a vitamin, but also with aquatics. So especially with salmon industry, what makes salmon pinky red? It's astaxanthin. They're white without it. And so they're actually feeding a sort of a semi-synthetic version of this astaxanthin currently to most of the farmed salmon in the world. However, there's now a massive interest in using the natural form because there are massive issues with the synthetic form. The isomer and kind of the stereochemistry is not the same. And so they're finding that the bioactivity is not identical. The natural form has far higher net bioactivity than the synthetic form. And so the community of astaxanthin producers and people that consume it and use it are starting to really shift and demand this natural form. So being able to increase this efficiency of production, because if you don't understand the process of how this pigment is produced really at a deep level, you can't really fully create a true efficiency in this production line. And that's true not just of my own opinion, but to those in Iceland that I talked to, also some of the other labs around the world, such as in Ireland and Israel and down in Hawaii and in Texas. They're all saying the same thing, that we need to increase the efficiency of this production, but we need to know what's going on first.
Great, thank you.
Any other questions?
I think Gary was the next one.
Gary, you need to unmute.
Okay, is that better?
Yep.
Yes.
Sorry. I have a question about the you use the stress marker ROS. Are there other markers that are relevant here with regard to how useful this would be as a nutraceutical?
Definitely. And so just as with any good study, you end up asking more questions at the end of it, I think, than you start off with, right? You open 17 doors by closing one. So also there's a lot of interest in RNS, so kind of reactive nitrogen species, as well as there is still some questions with ROS and kind of acting, well, where's the main production of the ROS? Because we think of mitochondria, right, with people and with cancer studies, but with things that are photosynthetic, you also have to think of the chlorophyll, of the chloroplasts. And so where is that coming from, as well as is it possible to control that? So even beyond just using kind of increasing the ROS of the cell, there's also increase in, basically there's about a list of like 50 different metabolites that shoot up when these dudes start insisting. So essentially they start getting really angry and really stressed out, and they just shoot into a little cell. And so there's actually quite a long list, but there is quite a lot of interest and research that I've seen into like RNS, ROS, that sort of thing.
Those are the benchmarks.
Yeah, those are like the big ones that definitely.
Thank you.
Okay.
Hi, this is Anne. I just had a quick question. Hi.
Yeah.
Could you just talk a little bit more about the methods you're going to use in terms
Sure.
of how you can like independently measure each of those, ROS and astrophin?
Yeah, so my main thing is obviously measuring the astaxanthin and measuring the ROS output. Astaxanthin is definitely the easier one. So I have been collecting preliminary data, and I'm in the process of actually working on this. And so I've been essentially freeze drying, hand cracking with a mortar and pestle, grinding these up, and then extracting the astaxanthin from this crude product of the dried. So that way you can get a direct measurement of how much astaxanthin is produced based on how many liters and the dry weight of the crude product is, so you can directly weigh the astaxanthin, as well as I am using HPLC analysis through USM, which is the industry standard at this point, as well as I did in Iceland, to be able to determine what product is actually produced. So to make sure that it's the correct carotenoid, instead of beta carotene, for example, or any other in this process, canthaxanthin. But also I'm able to tell what form of astaxanthin this is. There are three main ones. You want the free ester version of astaxanthin. This is the one that has all of the bioactivity properties. And so I'm able to measure that, determine it is the correct astaxanthin that is bioactive. And then for ROS, the main way that I have been quantifying and will be quantifying is using an ROS sensitive dye. The name's about this long, but it's frequently used in cancer research in order to measure ROS within cells that I've seen. And so using flow cytometry, a plate reader, and spectrofluoroscopy, and using fluorescent spectroscopy especially is wonderful here. I'm able to actually quantify the amount of ROS produced. This is the easiest way to do it. There are technically a couple other ways, but I've had pretty good success with this. This tends to be the industry standard. Mainly a lot of flow cytometry, to be honest. And so then from that data, I'm using a linear regression model to kind of compare and contrast these data points, especially within the different environmental kind of stressors, and see how that affects production.
Great, thank you very much.
Yeah, thank you.
Okay, thank you so much, Rachel. And with that, we will move to our next contestant, and that's Anastasia. Hi.
Hello. How are you?
Good. Are you able to share your screen?
Yes. 1 second. I'm working on it right now. Let's see. Is everyone able to see that?
Yes, perfect.
Fantastic. All right. So I'm currently researching multiple myeloma, and that's a cancer of plasma cells. It accounts for 13 to 15% of blood cancers and is the second most prevalent hematological cancer. In the bone marrow, these plasma cells proliferate rapidly, suppressing bone formation and increasing bone resorption. This results in very painful osteolytic lesions, as you can see here in the patient's skull and tibia. This causes bone fracture, bone pain, hypercalcemia, and nerve compression. Although there have been many therapeutic advancements over the last few years, patients with the cancer almost always relapse and develop a more malicious drug-resistant disease, resulting in an incurable cancer. Here in the bone marrow niche, along with malignant plasma cells, there are also adipocytes, which have been shown to secrete adipokines such as fatty acid binding proteins 4 and 5, otherwise referred to as FABPs. These FABPs have been shown to contribute to tumor growth, invasion, and survival. In my research, I decided to look further into the effects of FABPs. And my first test was to see if exogenous FABP4 or 5 had a proliferative effect on myeloma cells, or if the myeloma cells had a way to produce their own FABPs. In this experiment, I spiked in increasing doses of recombinant FABP4 or 5 into 2 cell lines, MM1S on the left and OPM2 on the right. These cells were cultured for 72 hours, and the results were analyzed via bioluminescent imaging. The results showed that external FABPs have no proliferative effect on either myeloma cell line, even with increasing doses. This suggests that myeloma cells must make their own FABPs. From that, I hypothesized that if we inhibit FABPs in myeloma cells, we could decrease proliferation and tumor burden, providing a novel mechanism to treat cancer. In my first experiment with this, I spiked in 50 micromolar of inhibitors of FABP4, 5, and a co-treatment into myeloma cell culture. The results were analyzed via bioluminescent imaging, and they showed that over time, FABP inhibitors significantly decreased myeloma cell proliferation. Given these results, I moved to an in vivo mouse experiment where I injected mice with myeloma cells and treated them with inhibitors of FABP4, 5, or a co-treatment over a 4-week period. My results showed that FABP4 inhibitor-treated mice had increased survival compared to the control, which you can see the control is the blue line here, and also significantly decreased tumor burden. I also included a bioluminescent scan of mice at day 24 and day 28. The luminescence on this scan resembles where the tumor is, and you can see that there seems to be significantly less tumor burden in all 3 treatment groups compared to the control or vehicle. To conclude, FABP inhibitors decreased myeloma cell growth over time in both in vitro and in vivo experiments. Furthermore, they increased survival in a mouse model. This supports the idea that FABP inhibitors could prove as a future cancer therapeutic. In the future, I also aim to test the combination method of inhibiting FABPs in myeloma cells, along with dexamethasone, which is a common chemotherapeutic drug, to see if this further decreases tumor burden and hopefully eradicates the cancer. Thank you.
Sorry, I went first last time and I realized that I'm supposed to go first on this one, so that's my fault. So that was great, Anastasia. So the FABPs, those are intracellular, correct? So are they binding lipids that are being stored in the myeloma cells, or are they helping with lipid uptake and oxidation? What is their function in these cells?
Yeah, so most likely they are helping with lipid uptake and reproduction, and then that is helping with tumor invasion, survival, and then growth as well. So they're utilizing these FABPs to help them sustain living in the microenvironment.
So these cells mostly use lipid and not glucose as a fuel then, do you think?
I think so, but it's still being researched as well.
Okay, thank you.
Of course.
Hi, I have a question.
Mm-hmm.
You hear me?
Yes.
Have you tried using BMA cells? Are there cell lines available that you could potentially co-culture?
Yeah, so actually some other people in the lab are co-culturing myeloma cells with adipocytes, and I have done some co-culture experiments by myself as well. And what we do with those is we get bone marrow donors from the hospital, actually, Maine Medical Center, and then we culture the mesenchymal stem cells and then differentiate them into adipocytes. So there's a lot that is going on with those, and something that we're also finding is depending on the donor and what the donor may be predisposed to or pre-exposed to, there are some variable effects on the myeloma cells.
Yeah, I was wondering whether you see proliferative effects that would occur, and whether or not the fatty acid binding proteins that you add would be enhanced by the presence presence of those cells, since those cells are present in the body.
Yeah.
Together with, so just a thought about that.
Yeah, no, fantastic. Thank you.
That's all I have.
Any other questions?
Okay. Thank you so much, Anastasia. So now we're moving to our third contestant, and I'm sorry if I say your name wrong, Boa.
That's great. You did great.
I did great. Oh, thank you. Okay. Can you share your screen, please?
Sure.
Perfect. Okay, and whenever you're ready, just tell us about your project.
Just a minute. Okay. Hello, my name is Adjoa, and today I'm presenting on a project which I have dubbed Simple Yet Impactful. All around the globe, diseases is a major concern. Its impact in terms of mobility and mortality, as well as economic impact, cannot be overlooked. In US, obesity alone costs over $140 billion every year in treating. The good news is we can take hold of certain modifiable factors like diet to control these diseases. The question that readily comes into mind is, if diet has an impact on our health, then what informs us in choosing the food we consume? The four major pillars for diet selection are availability, the cost involved, the taste, and the ease of preparation. Based on these pillars, it is known that 1 out of 2 people consume sugary beverage on a daily basis. This has increased our risk to several chronic diseases. Kombucha and kefir, a fermented sugary beverage, is known to meet these pillars. However, instead of predisposing us to several health concerns, these contain several health benefits that will be beneficial to us. These benefits have been tested and proven. They are known to help in diabetic treatment, have an antimicrobial and anti-diabetic and anti-inflammation effect, and also help in even treating cancer. Kombucha alone is known to contribute to over 30% of the food industry. This is very significant because it implies that if the health benefits can be sustained, then these health benefits will be reaching over millions of people. Kefir, on the other hand, is in its infancy stage. However, we believe it will grow over time because it has shown similar health benefits as seen in kombucha. A major concern is how this product is brewed. In both cases, they require a starter culture, which is very rich in good bacteria and yeast. At the end of the fermentation, they give rise to parent cultures that are used in the subsequent brewing. The brewing cycle goes on over and over and over again until the producer calls it done. With a repeated brewing cycle, the problem that we all glance at is there a change in the microbial community and the health benefits of a repeated brewing cycle? Hence, my research seeks to understand the microbial profile, the chemical composition, and how these change over 20 brewing cycles. In the end, we hope to come up with a standardization scheme that will be communicated to producers to help them in knowing when to call their products done. By simply coming out with a standardization scheme, we will impactfully help to preserve the health benefit that is attributed to this product to the consumer. Thank you.
Thank you, and we'll take a question.
Yeah, that was a great presentation, and I feel like this is the beginning of a very large study going into the different ideal outputs of different amounts of cycles and different conditions for you to select the best set of conditions for the kombucha, you know, the companies doing this industrially, what do you think the end result ideally is? What types of things are you looking for? And what amount of cycles or what variables are you going to dial to get there?
So for our initial plan before the COVID started, our cycles were running up to 20 cycles, and we decided to take DNA samples on 10, 5, generation 10, 5, 10, 15, and 20. And then in the end, we'll be comparing how these are changing. And then not just the DNA, we'll also be doing chemical analysis, because we have known from literature that it contains caffeine, ethanol, and other polyphenols that are of health benefits. So we were also going to do this by HPLC to know what was in it. However, during the COVID season, what we are doing now is wherever we are now, we are stopping at generation 10 for the kombucha, but the kefir takes a shorter time. So for the kefir, we are stopping at generation 20. And then after the season resumes, we'll be comparing our last generation 10 with the ones we store to see if there's any microbial differences. If there is no microbial difference, we'll continue the cycle. However, if there is microbial difference, we'll repeat the ones we store, and then that will be a replication of the first 10 that we have. In the end, we'll be communicating this to the industry. So our last part is essentially parameters which we are doing with the industry to get what they want to look at. Sometimes the kombucha has biomass being formed, and people do not like all those things. So in the end, we'll be communicating our results and our findings to the kombucha, especially to the kombucha industry.
Awesome, thank you. Just curious, how do you think it will change as you increase the amount of cycles, the microbial community? Did you catch that?
No, sorry, come again.
How do you think it'll change over time? I'm just curious, the microbial community with the amount of cycles.
So from what we know for the kefir, so one thing I should mention is the parent culture and the daughter culture that is produced is inseparable. So for the kefir, we believe the microbial community might not change that much, because you transfer both the parent and then the daughter culture into your next brewing cycle. However, for the kombucha, you just transfer the daughter culture that comes on top, and then that one will be the next parent culture for the next brewing cycle. So we believe there might be a vast variation over time for the kombucha. However, for the kefir, and even in literature, it's assumed that the kefir has less diverse community than the kombucha over several brewing cycles. And then one thing we are doing is, because there are several variations, we are trying to standardize our brewing. So the only thing that is changing in our experiment is the scobies or the culture that we are using, but everything is standardized. pH is standardized at pH 3 and then pH 4 for the kefir. So everything is standardized, so we can attribute whatever microbial changes to just the repeated brewing cycle.
Hi, you talked about collecting DNA to be able to evaluate the microbial population. Can you tell me specifically how you're going to be doing that?
Okay, so for the kombucha and the kefir, all of them will collect our DNA. You want me to talk about the DNA or the process?
Both, actually, if you could.
Okay, so for the kombucha and then the kefir, we'll be taking samples from both the culture, the started culture, and then the end product, which is the drink itself. And then we'll be extracting DNA from that. Then from there, we'll be doing amplicon sequencing.
For specific genes, or are you doing ribosomal RNA? What are you?
Yes, we'll be doing ribosomal RNA.
Okay, okay.
To look at the, right?
And have you done any of this so far?
No, so the whole thing is so sequencing is expensive. So we want to have as much DNA as possible, but with the whole COVID pandemic going on, we don't have much of the DNA collected to send it out. So we are hoping when things ease down and we go back, we get enough, and then we send everything out for sequencing. And then, but in the meanwhile, we are still collecting our DNA samples.
Is there any evidence from other studies or work, just from culturing or something, what this population looks like? Is it a very complex population, or is it predominantly one microbial source? What are you expecting out of that
So,
study?
so our study, we are doing both DNA, that is the high throughput sequencing, and both cultural based. And for the kombucha is known to have a high amount of lactic acid bacteria. And then the kefir is supposed to have a high amount of acetic acid.
Okay.
Oh, for the kombucha is rather acetic acid and kefir is lactic acid. However, with the ones we have done for the microbial plating on culture, this is very variable. We are having rather the opposite. So for the kombucha, we are having a lot of yeast. And then for the kefir, for the kombucha, a lot of yeast. For the kefir, we are having a lot of lactic acid. We will confirm this result after we ran the DNA to see. And then these results were done on shelf life basis. So we're doing every 2 weeks to see how it will change over 90 days. From literature, it is known, there is variable, that's what I'm saying, we are trying to standardize ours. It depends on when they took their sample, at what pH, where they had their culture from, and how they brewed it. Some use green tea, some use black tea, but we are using just the one that is done in the industry, which is black tea. So the community that has been shown in kombucha is acetic acid, primarily acetic acid bacteria. Acetic acid bacteria, and then for the kefir is lactic acid bacteria. But as I'm saying, there are variable species that has been found in each literature that has come up, depending on how they were brewed and where they had their culture, and then how long it was brewed for. Because over time, the sugar component is used up, and then you end up having a lot of acetic acid, depending on the duration. It normally goes between 7 to 30 days, but we are trying to just not use the day, but rather use the pH, because this is supposed also to be a non-alcoholic beverage. So we don't want it to go to 2.5, lower than 0.5 alcohol strength that is known in US. So we are trying to use a pH to standardize our brewing cycle, and we'll get the information to the industry.
Great, thank you very much and good luck.
Okay, thank you.
Thank you so much. And with that, we'll move to our next contestant, and that's Charlotte. Hi, Charlotte.
Hi. Let me share my screen.
Sure.
Okay, ready whenever.
Whenever you're ready, just start.
All right. What is this? Oh, okay. So proteins are vitally important for human health. They're responsible for our body's growth and repair. They protect us from illness as antibodies, and they make up 16% of our body mass. And these proteins couldn't be created without transfer RNA and their associated CCA sequence at the 3' terminus. And the CCA precedes the amino acid sequence of all proteins. And in most eukaryotes, it's synthesized by one tRNA nucleotidyltransferase, and I'm tRNA-NT for short. And Schizosaccharomyces pombe is the first eukaryote observed where 2 enzymes collaborate to create the CCA sequence. You have CCA1, which puts the 2 cytidines, and then CCA2 is the second enzyme, which adds the terminal adenosine. And so recently it's been discovered that defective tRNA-NTs can be linked to a multitude of different illnesses, including cancer, type 2 diabetes, and neurodevelopmental disorders. And so when these tRNA-NTs don't work properly, their associated tRNA can't function in translation, which impacts protein synthesis and ultimately renders them defective. And thus it's becoming increasingly important to understand how these enzymes work and the conditions under which they don't. And so that's what my research seeks to address. So a previously conducted study showed that by substituting the glutamic acid with phenylalanine at residue 189 in Saccharomyces cerevisiae, growth was inhibited at a restrictive temperature of 37 degrees Celsius. So then the alignment of the amino acid sequences for S. pombe and S. cerevisiae tRNAs showed that this glutamic acid at position 189 was also found in S. pombe tRNA. And basically for my research, I used site-directed mutagenesis to create mutant S. pombe CCA1 and CCA2 enzymes containing this glutamic acid to phenylalanine substitute at the position that corresponded to the one made in S. cerevisiae. And ultimately, successful S-Pombe CCA1 and CCA2 mutants were created by removing a BBS1 restriction site in the CCA1 and adding a BAGL2 site in CCA2. And the CCA2 mutant initially didn't work as seen on the agarose gel, but it was eventually successful once we optimized the annealing temperature. And the success of these mutants ultimately proves the capacity to create variable or viable variant enzymes with the potential to express these temperature sensitive phenotypes. And so this was basically just the first most difficult step of the process. So the next steps for this project will be to determine the activity and stability of these mutants using biophysical techniques. And because the variants correspond to those confirmed to have temperature sensitive phenotypes in S. cerevisiae, we strongly expect that the S-Pombe enzymes will also have the same reduced activity and altered thermal stability. And this outcome would be incredibly significant because temperature sensitive mutants provide a reversible responsive mechanism to lower the amount of specific genes being produced during any organismal growth stage. So, and you could do that only with changes in temperature. And so therefore creating these mutants is vital, not only for finding treatments to address illnesses caused by defective tRNA and Ts, but also for understanding how they function normally and why they go wrong in the first place. So yeah, thank you.
Charlotte, thank you very much. Appreciate that. Question really about the targeted site. So in CCA1, I know you only showed us a very small part of the sequence, but in CCA1 looks quite similar to the cerevisiae enzyme, whereas CCA2 actually doesn't share that same degree of homology at that particular site. Do you have other evidence that indicates that is a key amino acid, and replacing that will give you that same kind of temperature sensitivity?
Yeah, well, the lab that I'm working in centers research largely around the study of these nucleotidyl transferases, and they did a previous research study where they sequenced the CCA1 gene that encodes this tRNA nucleotidyl transferase, and they saw that a single guanosine to adenosine transition caused the conversion of this glutamic acid at specifically position 189 to change to lysine. And they didn't find any other nucleotide substitutions. So this kind of implicated that this change alone was responsible for generating the temperature sensitive phenotype.
I guess my focus is for the CCA2, the glutamic acid that you're replacing, doesn't seem to share the same degree of homology with the Saccharomyces cerevisiae enzyme as the CCA1 does. And so I'm wondering whether or not there's sort of other biochemical evidence that indicates that glutamic acid 164 is really the glutamic acid to be able to replace here. Yeah, I'm not really sure, but as far as I know, based on previous research, it's also related to the specific motifs that they're in. And my lab has been looking specifically into a relationship between motifs A and C for these amino acid sequences. And I'm pretty sure that glutamic acid is found also in the same motif C that, like when you line up the amino acid sequences for cerevisiae and then CCA1, CCA2, they're all in the same motif. So I think that's what a lot of it was. Excellent. Thank you. Appreciate that. Yeah, of course.
Thank you, Charlotte. So while we're on this slide, your mutation from the cerevisiae that led to no growth, that could just be a broad reduction in translation. But in your last slide, you talk about getting to the point of specificity where you can reduce certain tRNA's activity and target certain proteins. So how do you get from this general effect to a more specific targeted effect in the future?
Yeah, well, temperature sensitive mutants are really important for that because when you have like a typical temperature sensitive mutant, you have a drop in the level of activity for the gene product when it's expressed at a certain like restrictive temperature. And then any temperature below that is called like the permissive temperature. And that's when the activity is very similar to like the wild type of that gene. And so basically, because you have these two kind of polar ends, you can induce the phenotypes at restrictive temperatures and study the effects and see what happens. And then you can also just identify gene sets that are essential for different aspects of like biological functions. And detail like the understanding of how they work in the first place. And like a lot, I know one of the examples off the top of my head is they did this in order to find a lot of fundamental aspects about cell division using yeasts. And I think that you just use like live cell imaging, and you can see the way that these temperature sensitive mutants can change as they grow, and you can kind of control that as the researcher. So that's from my understanding.
Okay, thank you.
Yeah, of course.
Okay, thank you so much, Charlotte. Stop your screen share, that'd be great. And we are now moving to our next contestant, and that's Sarah. Sarah, are you there?
Yep.
Hi.
Wait a tick. Go. Let's share.
And just whenever you're ready, go ahead.
Okay. Hello all, I'm Sarah Holbrook from the Cox Lab at the Jackson Laboratory, and I'm excited to share one of my recent projects with you all. In collaboration with Nationwide Children's Hospital, we have conducted a pilot trial of 2 gene therapy vectors in one of our severe SMARD model mice. But first of all, what is SMARD? SMARD stands for Spinal Muscular Atrophy with Respiratory Distress. It is a rare childhood neuromuscular degenerative disease caused by recessive deleterious mutations in the IGH-MBB2 gene. It's characterized by neural degeneration starting in the distal limbs such as the feet and hands, leading to muscle atrophy and other side effects such as fatty pads. Before this occurs, though, typically within the first year of life, patients go through diaphragmatic paralysis, causing extreme respiratory distress, and usually necessitating the need for mechanical ventilation. Most patients die in early childhood, and there is no cure. That is, until maybe now. We tested 3 viruses on our severe SMARD1 mouse model. We have 1 virus that expresses higher levels of IgH-MAB2, 1 virus that mimics endogenous levels, and an empty vector as a control. I was blinded as to which virus is which, so I'll be calling them viruses A, B, and C with no order in particular. Our mouse model, C57 Black 6J NMD Cox EM3, or EM3 for short, has severe neuromuscular degeneration as displayed on the right by the lack of overlap between the red acetylcholine receptor channel and the green nerve channel. We also only live for about 19 days and show signs of paralysis even earlier than that. So, when we give our EM3 mice an intracerebroventricular injection of each virus at age day 1, what do we see? For our limited 8-week study, we saw an increased lifespan in virus A and virus C, as shown by the orange and green lines. Spoiler, virus B is the empty vector, so I won't be focusing on that virus going forward for this presentation. We also see increased muscle mass in virus A and C, with C being larger than A, but neither quite as large as the wild-type mouse. This is a cross-section of the leg muscle. And if we go deeper into the nerves of those leg muscles, you can also see that A and C are bigger once again than no treatment, with C being slightly larger. And if we go one more level deeper into the place where the nerve meets the muscle, at the neuromuscular junction, we not only see improved innervation, perhaps even occurrences of nerve sprouting, suggesting that the healthy nerves infected with the virus are re-innervating the muscles where uninfected or unhealthy nerves are not able to. So, in conclusion, the data I showed you, and countless other measurements I was not able to show you today, indicate that virus C, whichever that may be, is more effective, but both are much better than no treatment at all. And the severity of our mouse model allowed us to see minute differences in efficacy between the viruses, but our other collaborators working with less severe mouse models were not able to see. More pilot studies need to and are currently being run to see how this treatment in mice affects them in the long run, see if there's any overarching health concerns, the viral shelf life, and to see what is the optimal dose for best recovery. Thank you. And if you have any questions, you can contact me through email or Twitter or ask me now.
Thank you for that. I also am really happy that you added your Twitter handle to the end of that. I really like that. I was just wondering if you could talk a little bit about how you arrived at this as the mechanism of treatment when you were initially conceptualizing the study.
So we were technically reached out by Nationwide Hospital for this treatment, but the reason why we were already kind of thinking gene therapy would be good is because it's a homozygous recessive gene. Now, if this was something that was dominant, gene therapy itself would be a lot more complicated because you have to like knock down the bad protein and then reinstill like a good version of that protein. But because this disease is just caused by a lack of IgH-MAB2 or just has IgH-MAB2 that doesn't work all that great, reintroducing a healthy copy of IgH-MAB2 we predicted would help, and it has. We also tested the virus in wild-type mice just to see for any bad consequences of overexpressing IgH-MAB2, even when the mouse is already expressing healthy amounts, and we didn't see any overt phenotypes.
Can I get another question? Basically, I saw that the in vivo transduction was less sensitive than in vitro. And I was wondering if you couldn't take some magnetic particles and aggravate them with radio frequency to increase the blood-brain barrier permeability to get more transduction.
I'm trying to think. Our collaborators, so it's an AAV9 or AAV serotype 9. Yeah. The transfection is about 80%, which is probably why we see this. But yeah, I'm not quite sure like into the methods we can make it better. We're going to be having a meeting sometime soon. But we have also been testing a mouse model that isn't as severe. So when we inject these mice at day 1, it still takes a week or so for the single-stranded virus to activate. So this is almost kind of like a post-onset injection. So we have another mouse that's a Charcot-Marie tooth disease that starts showing paralysis and weight loss at 6 weeks. And we're doing P1 injections then to see how much of an impact we get. So like, even if we're able to save 80% of the nerves, is that enough to go all the way back to wild type levels? Like, is that enough to look like the wild type mouse? Or is there still some things that we need to work on for that transfection?
Okay, thank you. Very good.
Okay, thank you so much, Sarah. And if you could stop your share, that would be awesome. And with that, we will be moving to our next contestant, and that's Sydney. Hi, Sydney.
Hi.
Hi. Are you ready to share your screen?
Sure am.
All right. Okay, and just whenever you're ready, give it a go.
Hello, everyone. Thank you for having me. My name is Sydney Lang, and I'm an undergraduate from the University of New England, where the Department of Biomedical Sciences is studying a role for DOX7 in the crosstalk between the nervous and skeletal system. Osteoporosis is a condition of low bone density that renders bones vulnerable to fracture, affecting more than 54 million Americans. The prevalence of breaking a bone by the age of 50 due to osteoporosis is 50% in women and up to 25% in men. Osteoporosis is a disease that Americans are aging into without an understanding of how important the integrity of their skeletal system is to their overall health. Depicted in image A, you can see the two main players of the skeletal system: the osteoclast, or the reabsorbing breakdown cell, and the osteoblast, the building cell of the skeletal system. It is the balance of these that determine bone density. The balance between the building and breakdown of the skeletal system can be controlled by the nervous system as well. As seen in image B, the central nervous system sends messages to the bone cells. We refer to this overlap of communication as the bone nervous system crosstalk. One of the components of this crosstalk may be the DOX7 gene. a cell signaling protein which was first identified with the nervous system. Deletion of DOX7 in bone-building cells can result in a loss of bone density in mice using the Krelok's tissue-specific knockout process. It was an important discovery that bones are innervated with nerves, as you can see in the image by the sensory nerves. Does DOX7 function in the nervous system to control bone density? By studying the deletion of DOX7 in the nervous system and measuring the bone density in mice can contribute to answering this question. Understanding the role of DOX7 can lead to a better understanding of how bone metabolism works. A greater understanding of bone nervous system crosstalk, including DOX7's role in the process, could aid in the creation of new pharmaceutical remedies that don't cause bone loss, which is currently a problem. Understanding the role of DOX7 can also lead to more efficient treatments for osteoporosis as well. Lastly, I would like to thank Dr. Kathleen Becker and Talia Lazat for being amazing mentors leading this research. I would also like to acknowledge the College of Osteopathic Medicine, the Department of Biomedical Sciences, and the Office of Research and Scholarship at the University of New England. Thank you.
Philosophically, why would you suspect there be sensory innervation of the bones?
Why would I suspect that there would be sensory innervation?
There is, but what's the function of that?
So since that there's the crosstalk between the skeletal system and the nervous system, the skeletal system serves much more than just the structural support of the body. So the osteocytes, the osteoclasts and osteoblasts, react to the changes within the nervous system. So as there's sensory changes going throughout the body, it'll impact the way that the breakdown and reabsorption of the body, of the skeletal system occurs. Did I answer your question?
Yes, and how are you going to study the change in the DOX7?
So in the DOX7, using the Krelok's knockout system, this will be showing the, in the microarchitecture scans using the micro CT shown in image D, this can show the bone density change in the trabecular bone. And as shown in D, the normal DOX7 gene, and in the deleted bone, it shows that there's a difference in that bone density. So, and DOX7 being present in both the nervous system and the skeletal system will have that crosstalk make the change.
Good. Thank you very much.
Thank
Do
you.
you know if DOX7 is expressed in both osteoclasts and osteoblasts?
That is a very good question. I'm not certain that I can answer it definitively. I know that in traditional treatments for osteoporosis, they are targeting the osteoblasts, and I believe the osteoblasts, and this could be based on the DOX7 gene, but I can't confidently answer that question, but it is a good aspect to do more research on my part.
And in the context of your knockout here, beyond the sort of the bone density scans that you have you actually evaluated the sort of composition of the bone marrow to identify whether there's been changes in osteoclasts, osteoblasts, adipose, other kinds of cellular changes?
I'm not sure extending beyond the bone density of the scans that we were doing, but in addition to studying the osteoblasts and osteoclasts, studying the marrow in addition would be very beneficial to the study as well.
Thank you.
Okay, thank you so much, Sydney. And we're going to move to our next contestant, and that is Maria.
Thank you.
Hi, Maria.
Hi, how are you?
Good. So share your screen whenever you're ready. you can start your presentation whenever you're ready.
So hello everyone, I'm Maria Bina Lopez and I work at the Wheeler Lab at the University of Maine, studying the fungicidal synergistic activity of fluconazole and cyclosporine on Candida albicans. First, I want to do a brief introduction on Candida albicans and existing drugs. Candida albicans is an opportunistic pathogen that can cause mild use infection and it's capable of getting into the bloodstream and result in severe illnesses, as well as it can be life-threatening in immunocompromised individual. And this is why we aim to improve the current existing treatments available. So one of them is fluconazole, which is an azole drug that targets an enzyme in the biosynthesis of ergosterol, leading to membrane stress. This drug is fungistatic, meaning it inhibits the growth of Candida, but it does not kill the fungi. And also, with its use, it's developing drug resistance in Candida. We also want to introduce cyclosporine A, which is an immunosuppressive drug that exhibits antifungal activity by inhibiting calcineurin that acts on the fungus physiology. And we're introducing both of these drugs because it has been observed that there is a fungicidal synergy, so a killing of Candida between fluconazole and cyclosporine A. This can be seen in my in vitro experiment result. This is the scan of the plates after 24 hours. And if we look at the plate on the top, on the first row, we have Candida alone. And then from rows 2 to 7, we see Candida plus different concentrations of cyclosporine. And as you can see, the fungi is still growing and we did not observe any death. When looking at the second plate, on the first row, we have Candida plus fluconazole. And as you can see, there is less growth of the fungi. However, when we have both of the drugs present and the fungi, there is death of the fungi. So cyclosporine alone has no effect on the growth. Fluconazole only slows the growth, and both of them present cause Candida death. So we concluded the cyclosporine A converts fluconazole from fungistatic, so from only slowing the growth, to fungicidal, the killing of the Candida. So we want to understand how is this happening. So our goal is this, to understand how fluconazole and cyclosporine synergize to better treat Candida albicans infections. And how we're doing it is, right now we're looking at different time points to understand when the drugs are synergizing, but also we want to find the lowest concentration that is effective to be synergistic with fluconazole. In the future, we would like to look at the role of iron chelators and iron depletions, as well as the possible interactions with the bacteria Pseudomonas aeruginosa, and potentially looking at the interactions in vivo using zebrafish infection model. And all of this because it has the potential to help with efficient use of these antifungal drugs to treat the infection, as well as to prevent drug resistance. So thank you all for your time, and I would want to acknowledge the Wheeler Lab, Dr. Wheeler, Dr. Hattab, and the University of Maine. And I'll be happy to answer any of your questions.
Hi, Maria, this is Anne. Nice presentation. So these experiments have been done before. And so this, there has always been shown this added effect of fluconazole with cyclosporin A. And so do you have plans to look at some of the genes that might be important in this synergy between these two drugs?
Thank you for your question. I think that would be very interesting, but at the moment we're trying to figure out what is exactly happening with two drugs. So what is making them being able, when combined, to kill the fungi? So that is what we're trying to figure out first. But I think it would be very interesting to look at the genes as well.
As you move to the zebrafish model, what types of other factors do you think you'll want to account for, consider?
Yeah, so here I wrote it briefly how we wanna, when we use the zebrafish infection model in the future, we would wanna look at how if the synergy is happening in the zebrafish. And we're also unsure of the effectiveness of the cyclosporine A on fish. So we don't know how it's gonna act. And potentially we would also look at the host response to the infection.
Okay, thank you so much, Maria. And we're going to move to our next contestant. So Maria, you can stop your share. Thank you. Our next contestant is Caitlin. Hi.
Hello.
Hi, we can hear you. So whenever you're ready.
So my name is Caitlin Stieber, and I'm a PhD student in the lab of Dr. Lucy Liao at Maine Medical Center Research Institute. And my project focuses on understanding how perivascular adipose tissue, or PVAT, the fat that surrounds most of the vessels in your body, affects cardiovascular disease. Cardiovascular disease is the leading cause of death in the United States and is tightly linked to obesity. And because PVAT sits on the vessel wall, this provides a cellular link for us to be able to study these diseases. And one of the ways we can study this is through studying fat differentiation, the process where PVAT progenitors become pre-adipocytes and then become mature adipocytes that sit on the vessel wall. In healthy persons, PVAT secretes factors that decreases vasoconstriction and proliferation in smooth muscle cells, maintaining regular blood flow. However, in obesity, when you have an expansion of this fat pad, what is secreted changes, causing an increase in vasoconstriction and proliferation of the smooth muscle cells, narrowing the vessel, which limits blood flow and contributes to disease pathogenesis. But what if we had a way to control this differentiation? One of the molecules we are looking at to do this is called RAB27A, and it is expressed in both mouse and human PVAT. Additionally, our lab found that when you knock down RAB27A or you limit its expression in adipose progenitors, when you then differentiate this adipose into mature adipocytes, you see less lipid formation, which means that RAB27A functions in controlling adipogenesis. And we want to be able to study this in a variety of human cardiovascular diseases. So the way that we do this is we take perivascular adipose tissue from humans undergoing open heart surgery, and we are able to isolate PVAT progenitors from those samples and then form 3D models, cell models of adipose called adipospheres, which are shown here. And these are advantageous over traditional 2D models because it more closely mimics cell-to-cell connections that we see in the body. And additionally, we are able to easily co-culture cells in this system. So you have endothelial cells and adipocytes in this particular system. And eventually what we want to do is expand this to co-culture PVAT and smooth muscle cells together from two distinct cardiovascular populations. And that way we will be able to model perivascular adipose tissue sitting on the vessel wall and be able to understand this signaling and how it changes in a variety of cardiovascular diseases. I would like to thank my lab, my funding sources, and I can be contacted at the email address below.
Thank you, Caitlin. This is Ann. Can you tell me a little bit how you're going to validate this 3D model that you've created of these adipospheres?
Yes. So far we've started validation in mouse models of PVAT. So we're able to excise and do the same process with the mouse tissue as we are with the human tissue. And there we have been able to show expression of RAB27A in those adipospheres. So we know that it is expressed in the tissue and that we can get this expression in the model we propose to use. So we will continue using that, and eventually we'll look at RAB27A expression in human adipospheres. But in terms of controlling for this, the two cardiovascular disease populations we're planning to use, one we expect to have arterial disease and one we expect to not have arterial disease. And that will basically be our control in terms of the PVAT modeling, where the PVAT from the non-arterial disease patients, we expect that to behave more like healthy PVAT as compared to the disease PVAT that will come from the patients with arterial disease.
Thank you.
Hi, Caitlin. This is Stephen. I'd like to go back to your knockdown experiment quickly. And while you see obviously decreased expression of your RAB27A, you still do see some adipocyte development in those cultures. Do you think that that is a technical issue with regard to the knockdown process, or do you think that there are some cells that are able to develop even in the absence of RAB27A? What do you think is going on there? Is that sort of an experimental issue, or is it really a differential cell pathway?
So the knockdown is not complete, so that is contributing to this. But additionally, we hypothesize that how RAB27A is controlling this is that it is secreting different adipokines that may be contributing to this increase in the amount of PVAT. So it's possible that something is happening there as well.
Great, thank you very much.
Thank you, Caitlin. Very nice.
Thank you.
And we are now moving to Adrian. Did I say your name right, Adrian?
Yes, just a minute.
Sure. Okay, we can see your screen, so whenever you're ready, just start talking.
All right. Hi, my name is Adrian Tracy, and for the past year and a half, I've been working in the lab of Dr. Jose Fernandez-Robledo at Bigelow Laboratory for Ocean Sciences. As many of you know, oyster aquaculture has been a booming industry in Maine that has brought lots of economic benefits to our state. In addition, oysters have actually been linked to cleaning up organic and inorganic pollution in our coastal waterways. That being said, there are over 32,000 predicted genes in the eastern oyster, but we really don't know what most of their functions are, given that many of these genes are novel. However, people have started to use these genes and the oysters as model organisms. For example, shell formation and shell matrices have actually been able to stimulate human bone cell growth. In addition, people have started to study oyster immunity because they have an innate immune system and can quite literally live in a sea of pathogens without any antibodies. That being said, we need to start to understand the function of all of these oyster genes. So we decided to develop genetic tools in order to study these oyster genes. We used plasmid DNA with green fluorescent proteins in conjunction with chemical dendrimers, which are a chemical compound that binds to DNA and transports it across the cell membrane. We put these in solution with oyster blood cells and tested gene expression with flow cytometry and confocal microscopy. From there, we were able to see successful expression of our genes in the oyster blood cells. You can see here with the confocal microscope image, the green fluorescence in the oyster blood cell. In addition, we were able to quantify the number of cells that exhibited fluorescence using the flow cytometry. We were also able to verify that our genes were being expressed by using antibody tagging, as you can see with the co-localization of the red fluorescence and green fluorescence in the same cell. Now, we are the first lab to be able to make this method work and express genes in oyster blood cells in vitro. This is just one of the first steps to decoding the oyster genome, but there are so many practical applications. For example, we hope to begin to develop a cell line with oysters, which means we would take some of the cells from the oysters and begin to grow them and propagate them in the lab. This would hopefully allow us to start industries such as cellular aquaculture. There are many other practical applications to our method, and I would like to acknowledge Bigelow Laboratory for Ocean Sciences and Colby College, as well as Jose Fernandez-Robledo's lab. Thank you.
Thank you, Adrian. So if you're expressing genes in these blood cells from the oyster, how is that getting you more information about the function of those 32,000 genes that the oyster already expresses? So which genes are you putting in?
So what we have is 32,000 predicted genes, and the plasmid that we used actually has a multiple cloning site. So we can basically cut and paste any of those 32,000 genes into the plasmid, and it will be tagged with green fluorescence. So we can see how those genes function in the cell, where they're expressed, and when.
Okay, but not all of them would be expressed in the blood cells, right?
Not necessarily all of them, but we may be able to use this method in other cells in the oysters as well. We would have to do further research, obviously, but hopefully this method would be applicable to other cells.
Okay, so this is the proof of concept step, and then you take it from here. Okay, great. Thank you.
I just want to ask you a quick question. Do you think it's possible to increase the fusion capability or transfection capability with things like PEG, or the other thing I've seen, you polyethylene glycol, or the other thing I've seen used is nanoparticles to help get foreign molecules into cells?
I'm actually not aware of any of those techniques. I don't think that we use them in our lab. I might be wrong, but in the past we've used electroporation, a physical transfection method. And from that, we didn't actually do any viability tests, but we found a lot of cell death and basically a lot of cellular damage under the microscope. And so our method gives us what we think is, it allows a lot more of the cells to remain alive after transfection. I'm not entirely sure about those other methods, and I don't think I can really speak to that, but that's really something we should look into. Thank you.
Sure, they use it with viruses primarily. I've also used centrifugation to get viruses into eukaryotic cells, and that's helped quite a bit. But looking at the defense mechanisms and the prophenol oxidase system, that I've worked with the silkworms and tarantulas to detect gram-positive toxins. And we're right now running into a problem with limulus, which is used to detect endotoxin in the pharmaceutical industry. And the Charles River just came out using tarantulas, but it's a poor source. And I'm thinking if you're looking at pyrogen contaminations in the pharma industry, the hemolymph from oysters may be a worthwhile alternative as an economic outlook.
Yeah, that seems like a
quite given what
great
you're doing, I'm sorry, but thank you.
Yeah, thank you.
Okay, thank you so much, Adrian. And last but not least, we have Victoria.
Hi.
Hi, Victoria. Whenever you're ready.
Okay. Hello, everyone. My name is Victoria Zuko, and today I'm going to present the work I did at the Prudowski Lab at MMCRI during this last school year. And it is about the unconventional effects of the trinexamic acid. The trinexamic acid is a common drug administered by emergency physicians to patients.
I'm really sorry to interrupt, Victoria, but I think you're sharing the wrong screen. Maybe we can't see your slides. We see a picture of you.
Oh, I'm sorry. I'm sorry.
No. I just wanted to make sure we could see them for you.
Okay. So the trinexamic acid is a common drug administered by the emergency physician.
I still can't see it. Can other people see her slides?
No, Victoria, you may need to stop the share and start it again. Just make sure the slides.
That's weird.
It does say PowerPoint all the way up, but we can see your picture and then there's like a city or something in the background. And if it doesn't work, we can pull it up too.
Let me try again then.
Yeah, just try again.
Okay, so. Can you see it?
No.
That's weird. I'm sorry, I don't know what's going on. I am sharing, but
Okay, so maybe Brianna, can you, oh, I think we can see it now.
oh yeah, can you see it now?
And don't let that stress you out. You can just start again as if nothing happened.
Okay, I'm sorry.
No problem.
Can everybody see now?
Yes.
Okay. So tranexamic acid is a common drug administered by emergency physicians to patients that are suffering from hemorrhage. When there's the loss of blood, we have to try to prevent the fibrinolysis, which is the breakdown of fibrin in clots. TXA is a very efficient inhibitor of the maturation of plasmin to plasminogen. Patients who are treated with TXA present with less inflammation and edema, and they recover faster. The goal of our lab is to answer the following question. What else is TXA doing besides preventing fibrinolysis, reducing edema and inflammation? So previously, it has been shown that TXA prevents the increase of the markers of cell damage. The main one is mitochondrial DNA that leaks out of the cell in case the cell is damaged. Our group also observed that the TXA is increasing the respiratory capacity of the cell and the synthesis of ATP. And my task in our lab was to figure out what else was TXA doing besides that. The experiment we conducted used endothelial cells and consisted of one control group and 2 different concentrations of TXA, similar to the clinical use. The experiment was done by fluorescent staining with a marker called, I forgot the name, I'm sorry. Our group also observed that you can see on the second image that the mitochondria is better and bigger in shape. We can also see that the data underneath was by Western blot, see that COX-4, which is a major marker of the mitochondrial biogenesis, was increased. And we also looked at phospho-AKT and PGC1 alpha. We used a method called DARTS to find out other molecules that TXA targets, and our major detection was pyruvate kinase M. This molecule is known to downregulate the mitochondrial biogenesis. And based on this, we hypothesized that the tranexamic acid could inhibit the activity of pyruvate kinase M and also increase the mitochondrial abundance and respiration. This hypothesis will be pursued in the future, and I would like to thank you guys for all your time and patience with the problem with the slide sharing. Thank you.
Thank you, Victoria. Don't worry about that. We've all had technical difficulties before. My question is about your findings related to the mitochondria. So the MitoTracker Red, I think that's mitochondrial activity. Yeah, so that's mitochondrial activity, and it looks like some of your markers down below are also increased at different concentrations. So how could these mitochondrial effects relate to what's seen in the clinic with how this drug works? I think you had edema, inflammation, things like that being reduced. How do you think the mitochondrial target could relate to those clinical observations?
So we can see that TXA increases the mitochondria biogenesis. So in case the patient has trauma, the mitochondrial cells would the mitochondria and inside the cells would be damaged, and you would see on, for example, on the first image, that's normal. So that's a normal cell. And even if you treat a normal cell with TXA, the mitochondrial size and the stain in red is darker, I would say. So it it shows us that it increased the mitochondria, and mitochondrial respiration is, how can I say that? It's like a marker of how well the cell's doing. So TXA helps with the cell that are damaged and those even that are not damaged.
Can I ask you how you measured the respiration in the mitochondria?
So the respiration, that is the seahorse experiment, and I was not in the lab at that time. I'm pretty sure that was 3 years ago, so I'm not 100% sure how that was done.
Okay, thank you.
Any other questions?
I think Gary had the last one, if you had one, Gary.
Find any correlation between the, or measure for correlation between the mitochondrial release, increase in release, and oxygen uptake. The oxygen increased, I think you mentioned something about that. Is that right?
I can't hear you, I'm sorry.
You mentioned something about the oxygen increase in the mitochondria. Did you look at the relationship between those two things?
I'm not sure we looked exactly at the oxygen intake, but we did make an experiment to try to stress the cells in a hypoxic situation. And we treated it with TXA, and it increased a lot the survival of the cell. And it was
I mean, is that due to mainly, oxygen, additional oxygen for the cell? That's my question.
For respiration?
Yes.
I don't know if we looked at that. I'm sorry, but it's something we should.
Thank you.
Okay, thank you so much, Victoria. I think that concludes today. I would like to thank everybody who devoted this 1 hour and 22 minutes to us today. You did a great job. Thank you to all the judges. And normally now we would go off and have a reception, maybe some kind of happy hour and networking, and then we would announce the winners and all clap and be happy and celebrate. But unfortunately, we had to do it in a different way. However, the awards are still the same. So I want to wish good luck to everybody, and you can go and do your happy hour on your own, I guess.